in addition to the solid tumor strategy we're going after that we've articulated, we now have data that gives us conviction to go after KRAS-driven tumors, which is a whole new set of opportunities that, again, we don't think any other TROP2 ADC is looking at today because their payload, we don't believe, is as effective in these KRAS-mutant tumors as our payload.
Welcome back, and thank you for joining us for another Virtual Investor What This Means segment. My name is Janine Thomas. I am CEO of JTCIR, and I will be the moderator for this segment. Today, we're excited to welcome Akari Therapeutics back to our Virtual Investor platform, and I am very pleased to be joined by Abizar Gaslightwala. He is Director, President, and CEO of Akari Therapeutics. Welcome back, Abizar.
Thanks, Janine. Thanks for having me.
Always a pleasure to have you. Before we get started, I just want to inform our audience that Akari Therapeutics is listed on NASDAQ and trades under the ticker AKTX. And during today's discussion, the company will be making forward-looking statements, and I encourage everyone to view the company's website at akaritx.com or the SEC's website for their latest filings and information. So for our audience, a little bit of background. Akari recently announced preclinical data demonstrating therapeutic potential of the company's product candidate AKTX-101 for hard-to-treat KRAS-mutant pancreatic cancer. So Abizar, first of all, congratulations on this exciting data. To dive right in, can you provide the rationale for a summary of the study you conducted?
Sure. Yeah, great, Janine. Thanks for having me here. Really excited to go through this data and give some more context to the press release we recently did. So just to kind of talk about our splicing modulating payload PH1, it all starts there. We know we have some really unique biology with the splice modulating payload. What we're finding is every day we look further and do more experiments, we're finding something else new. So in addition to killing cancer cells and creating unique immune activation of cells as well for the immune system to attack cancer, we're finding out affecting the splicing biology also has an effect on some oncogenic drivers, notably one of the most prevalent ones called KRAS. KRAS is an oncogenic driver in about 30% of all many solid tumors. Think about lung, think about colon, think about pancreatic. It's also in others. And so what we found in the past is in some preliminary experiments, we actually found our splicing modulating payload PH1 had an effect on a very specific subtype of KRAS mutants called G12V. There are a number of different KRAS mutants. This one, G12V, is more prevalent in colon cancer and pancreatic. It's also present in lung at a lower percentage. And so, we found this initial early filing. We actually filed it in one of our early patents for our payload. And we said, well, what's there to this splicing modulating payload? This isn't something we expected to actually affect KRAS inhibitors. We didn't actually create a payload that inhibits KRAS as a pathway. So it's a little unusual in this finding. And this is, again, where the biology, the more we do, the more we find. And so we repeated that experiment, that initial finding that we put in a patent. We repeated that experiment recently with our TROP2 ADC AKTX-101. And what we found were some pretty striking results in multiple pancreatic cancer cell lines in vitro.
These are very compelling. Appreciate that background. So to break this down even further for investors, can you highlight the key takeaways from the outcome of this study and why these data are important?
Sure. So let me also take a high-level step back up. Pancreatic cancer is one of the deadliest cancers we still have today. And there has been relatively little advances. There have been new products, but you still look at what we call the median overall survival, where 50% of the patients survive more and 50% survive less. That number is only 1.4 years. Imagine being diagnosed with a cancer where your median survival, which means 50% of people or less, is only 1.4 years. If those of you know anyone affected by pancreatic cancer, it's a very deadly metastatic cancer, still in need for many good options. So you start there and say, wow, how do we actually go after these patients? Well, in particular, these KRAS mutation patients, that means their cancer is even deadlier than the number I just said. KRAS is this very, very bad, what we call oncogenic driver. It causes the cancer to be even worse. So imagine even a worse outcome than what I said just now, the 1.4 years. So what this means is when we looked at specific pancreatic cancer cell lines, these cell lines that are kind of the histology or the makeup are of pancreatic tissue, they're cancerous and they have certain properties. We actually tested our ADC, the AKTX-101, and because it binds to this antigen called TROP2, TROP2 is also expressed in pancreatic cells. So we said, okay, we think there's a theory for why it should work, our payload should work, and it did. But what we found was really surprising. Not only did it kill the cells with high potency, and we measure potency in the what we call nanomolar range, which is a billionth of a mole. It's a really small quantity. So we had some really very potent results with that, with our ADC. What we also did is we did a comparison against a traditional KRAS inhibitor, a molecule that was developed to be a KRAS inhibitor, whereas we're just developed as an ADC payload that's much more broad. What we found is pretty striking. One of the leading, what we think potentially KRAS inhibitors called daraxonrasib. It's a molecule being developed by a well-known company called Revolution Medicine, and it's in phase 3 trials for pancreatic cancer. In this experiment, we actually found in 2 of the cell lines we tested, we found that our TROP2 ADC was more potent than daraxonrasib, which is a traditional or a very structured KRAS inhibitor. We didn't expect this. Not only are we seeing efficacy in vitro, we're seeing more potentially better efficacy or potency than a KRAS inhibitor that's in phase 3 testing right now. So what this means is this opens up a whole new set of opportunities. In addition to just going after cancer cells that express TROP2, we might be able to go after KRAS mutant cancers. Again, that is one of the highest unmet need cancers out there. It's a unique thing that other TROP2 ADCs may not have access to, and it's all because of our payload. Because our payload works very differently than all the other TROP2 ADCs, and we're finding efficacy in these KRAS mutant tumors.
Gotcha, Beezer. This is so important and so promising for the pancreatic cancer patients community, but also beyond. And if you think about the ADC space, there's so much attention, so much excitement. As you know, this is a What This Means segment. What do these data mean for your ADC development strategy and next steps?
Sure, it just adds more conviction and confidence into the path we're in. So I think as we've disclosed earlier, we're taking AKTX-101 into potential clinical testing. We have a plan. We're doing IND enabling studies. We hope to file our IND or clinical trials application by the end of next year and start our clinical trial with a focus on urothelial/bladder cancer and some other solid tumors. That is our initial kind of beachhead or area to start our clinical trial testing. What this data says is that, wow, we can potentially even go beyond that. We can go after things like pancreatic cancer, but specifically KRAS mutant pancreatic cancer, or even lung mutated KRAS cancers, which again is a big unmet need. So in addition to the solid tumor strategy we're going after that we've articulated, we now have data that gives us conviction to go after KRAS-driven tumors, which is a whole new set of opportunities that, again, we don't think any other TROP2 ADC is looking at today because their payload, we don't believe is as effective in these KRAS mutant tumors as our payload. So again, it gets back to our central thesis for why Akari is focusing on novel payloads. Our payload PH1 is very unique and it leads to different outcomes for patients potentially.
These are very exciting. I'm going to use your words. These data are wow. Thank you for sharing. Thanks for breaking this down. I don't know if the press release that you put out really went into this much of detail or just gave the context that is so important that came out of this discussion. So thank you so much. Congratulations on the data, and certainly can't wait to have you back to hear more as the story evolves. So with that, this does conclude the Virtual Investor What This Means segment featuring Akari Therapeutics. And I'd like to thank Abizer for joining us today. As a reminder, Akari Therapeutics trades on NASDAQ under the ticker AKTX. And if you like what you saw today, I encourage you to visit akaritx.com for more information on the company, to sign up to follow the company to receive their alerts, as well as follow their social channels to stay current on the latest information. And you can also visit virtualinvestorco.com for our latest segments and events calendar. Abizer, again, congratulations. Can't wait to have you back and hear more of this exciting program. And I wish everyone a great rest of your day.
Thanks, Janine. Thanks for having me.